this class. But let me just explain that very briefly. The input to building a profile is a multiple sequence alignment, abbreviated MSA. And when you run SciBlast, what's happening is SciBlast, you have the query, and then SciBlast, so that's some length, and SciBlast retrieves homologs, that's the first iteration is blast, and the homologs might align partially, and some of them might align over different regions. Then these guys are aligned to the query. This is called a master-slave approach. So you can say that in Amsterdam, right? You might not be able to say that in parts of the US. It's a master-slave approach. The query is the master and all the sequences are aligned to the master. It's called a master-slave approach. A hidden Markov model or profile also uses a master-slave approach. The profile of a hidden Markov model is the master. Everything's aligned to it. That gives you an alignment of things to the query. That multiple sequence alignment then is used to create a profile. What happens is that every column in the alignment, you might have 1,000 sequences in the alignment, and some positions are gapped over some number of sequences. So you have some number of amino acids, and you turn that vector of amino acids, N1, N2, there are 20 types of amino acids. So how many alanines did you see? How many cysteines did you see? So they're typically ordered alphabetically. You turn that vector of amino acids into a probability distribution of the amino acids. Now, if you use the raw frequencies, let's say you have an alignment and you have exactly 4 sequences, and you use the raw frequencies, so you have 3 L's and 1 I. If you use the raw frequencies, that's the maximum likelihood result. That says that you're going to have 75% probability of L, 25% probability of I, and 0 probability of every other amino acid. So that's if you use the raw frequencies. That's not a good thing to do if you're trying to generalize your profile for distant homologs, because you're only going to recognize what was in your data set. So what happens when you're doing a profile construction is you're going to use either a substitution matrix. Does anyone know the name of the substitution matrix that NCBI uses for BLAST? So a substitution matrix says which amino acids substitute for others. And the one that's used most commonly is Blossom 62. And this is from the Hennicoffs, Stephen Georgia Hennicoff in Seattle at the Hutch, part of the University of Washington, basically. So Blossom 62 is a substitution matrix. You might also use Dirichlet-Musso densities. That's typically used in Markov models. This is beyond the scope of what you need to know today. But the thing is that they allow you to include similar amino acids. So L and I are most similar to what other amino acid? Anyone know? Valine. So leucine, isoleucine, and valine are very similar to each other. So if you see leucine and isoleucine, you're probably going to see valine among some homologs. So they're going to include similar amino acids, and all amino acids will have some probability. But sequence weighting comes in when you try to allocate some weight for each sequence so that you don't let an overrepresented group dominate. So if this were using politics as an example, if you had in a group, let's just pretend, of all the students here, let's pretend we know it's not true, that 98 students all came from one small town, and you're all white and upper middle class, and then 2 students out of 100 came from 2 little towns somewhere else, and now you wanted to, and from very different backgrounds, and you wanted to have a vote on what you should do. You'd want to have the 2 students from the smaller towns have more weight than their numbers dictate, because you don't want the large groups to dominate. So if, for instance, in your group you had human, chimp, gorilla, and that would be cow, human, chimp, and gorilla are so closely related that you don't want to let them have 75% of the weight, or you're only going to recognize the hominids. So you're going to say, maybe we have 50% of the weight go here, 50% of the weight goes here, and that gives the outer group a little bit more power in the vote. So that's just at an intuitive level how it works. The next question was about complexity. Some algorithms are very complex, some problems are very complex. So from a computer science perspective, computer scientists are all, and I'm a computer scientist, so I can speak from personal experience, are always looking for how to improve or reduce the complexity of a task, or come up with a better solution for a task, either make it faster or make the results higher precision or higher recall. There's so many problems in computational biology. So if you're coming from that background, you want to say, these are the tools I have, what problems are amenable to these tools, and for which some type of speed up or some improvement in the performance is desired. And so if any of you, I know there's at least one person who has that question, have that question, you can come to me afterwards and we can talk more about it, because it's a very interesting question. Domain shuffling and promiscuous domains. Promiscuous domains are domains that are found in many different combinations. So kinase domains are an example of a promiscuous domain. When you have a domain that's found in many different combinations, it's very easy for it to show up in hits for which the homology is restricted to a local region. Domain shuffling happens typically when the domain is on a single exon. And exons and intron boundaries would make it easier for that domain to be merged with another combination of domains, so you can get these different domain architectures or domain organizations. Earlier I was talking about the similarity between functions. So we have plant and insect defensins in the same superfamily as scorpion toxin. And it would be really handy to have an ontology that actually recognized that similarity, that relationship. So the question is, does the gene ontology have a way of explaining what aspect of the function is in common between a scorpion toxin and a plant defensin? Is there a way of describing it? Now we know that the plant defensins and the scorpion toxins operate in a way, there's some aspect of the mechanism of their function. They permeabilize membranes. So we could say the function of permeabilizing membranes is in common between the two, between plant and insect defensins and scorpion toxins. Is that represented in the gene ontology? I don't know. But it'd be really handy if the gene ontology or some other ontology represented the similarity between functions by using SCOP or other databases that have encoded the structural similarities between proteins. This is kind of this concept may not be very obvious to you. Does it make sense to anyone here, what I'm saying? No. Okay. All right, I'll give you a better example. You guys have heard of Prozac. You've heard of selective serotonin reuptake inhibitors. So they target proteins that include G protein coupled receptors, GPCR, G protein coupled receptors. These are 7 transmembrane receptors. 7 transmembrane receptors. They include serotonin receptors, dopamine receptors, opioid receptors, cannabinoid receptors, odorant receptors, and many more. So all of these G protein coupled receptors recognize different ligands. They're related by evolution. That's easy to show from sequence similarity searches. And the functional similarity is that they are 7 transmembrane receptors for different ligands. You can describe that as being what defines them all as a class. I'm sure you're going to see that relationship encoded in the gene ontology. No question about that. Because you can describe this class as 7 transmembrane receptors that are part of the G protein coupled receptor signaling pathways. So the ontologies will represent this family relationship at a high level, but these subgroups, serotonin receptors, are different from dopamine receptors. So that's going to be part of the ontology. It's going to separate them out when you get to that lower level. That's the way SCOP does in families. Superfamilies, folds, superfamilies. All of these are types of ontologies, and they're represented as graphs. So the question is, can you take the information in structure that says, for instance, in SCOP, that these proteins, some proteins are related, like the plantophensins and scorpion toxins, and say there must be some way of describing the functional similarity between them. It's a high-level functional similarity. Does the gene ontology provide that for the defensins and the scorpion toxins? That I don't know, but that would be a really good question to try to solve computationally somehow. But that's actually really hard, so I just wanted to touch on what I meant by that. Profile pollution versus profile drift. Profile pollution happens when you include a false positive. So FP is the shorthand for false positive. If you include any non-homologs in your profile, they pollute the profile. And then the subsequent iterations, you may detect more and more of these non-homologs. Profile drift can look the same, but it's not necessarily pollution. Profile drift happens when your query, we can use this example. Let's say your query is part of its odor receptor. But odor receptors are related to other G protein coupled receptors. And if while you're iterating BLAST or SciBLAST or hidden marker models, you get into the serotonin receptors and the dopamine receptors and other receptors, then these guys may dominate the profile. And the odor receptors may not actually be recognized by the profile because they start being dominated by these statistics. So they're all related, but it starts to focus on this side of the family. That's called profile drift. It just goes over time, over each iteration, into a different part of the family space. And there are ways to address that problem. If you have more questions, just either you're going to raise your hand and say, hold on a second, explain that.
So,
Yeah?
Profile pollution and profile drift are not the same. Profile pollution causes profile drift.
No. No. Profile pollution means you have included a non-homolog. Profile drift you can get even if you don't include a non-homolog. But if you have included a non-homolog, then profile drift, let's just put it this way, profile drift includes profile pollution. So there's two ways of doing profile drift. One is all stays in the family, and this one you get non-homologs. So in each case you have profile drift, but if you've included non-homologs, that's called pollution. And if you haven't included non-homologs, then it's just drift. Does that clarify it?
Yeah, but why would profile drift if it's still in the same family?
Well, profile drift could stay in the same family if you have a very large family. If the, for instance, in SciBLAST, the program defaults are to show you the top 500 sequences or so. So the top 500 sequences might be dominated by a different part of the family, and then you're not going to be seeing the members of the family that we started off with. Does that answer the question? Okay, so now let's continue. I think it's not a problem with me finishing everything. So at Uniprot, there's a lot of tools to help you use Uniprot. So getting started here, they get a lot of money from all over the world from funding agencies, including in the US. And at the National Institutes of Health, I was one of the review panel that evaluated Uniprot. They get millions of dollars in the US every year. And they are required to make their tools very user-friendly. In fact, they're very easy to use. So I want to encourage you to use them if you haven't actually. So the KCNA1 human, so the structure of these identifiers, you have a gene name, then there's an underscore, and then there's the species. So the species is not spelled out completely. So it's typically 5 characters, but sometimes more. But the first 3, and then the last, so DROME, does anyone have an idea of what that might refer to in the species? It's Drosophila melanogaster. So it's the first 3 characters of the first part of the name, and then the last 2 characters, first 2 of the second part of the name. Mouse is just going to be mouse, rat is just rat, but otherwise you'll see very often these 5 characters, first 3 and then the next 2. So this is part of what was called SwissPro. Here you'll see it's labeled as curated, and it's called reviewed here, so the status is reviewed. And there's evidence that the protein exists at the protein level. So some genes are hypothetical. They come up in gene identification systems, but there's no evidence that the gene actually encodes a protein. It may be a pseudogene, for example. So the gene name is KCNA1, the organism is Homo sapiens, and you can actually go get the sequence and many other aspects of it from the same page. There's a lot of gene ontology data. So for instance, for molecular function, you see all these different aspects. You can see the source here is UniPro KB, this is kappa. This is you've heard of CASP for protein structure prediction. Kappa is for function prediction. So automatic methods for protein function predictions, that's kappa. There's all these different sources for the annotations, and they are used to label proteins with gene ontology annotations. And then you have the biological process, and there's many different biological processes that come up here. And you can go to Quick Go, which gives you the rest of the annotations. So they're just showing you the first number of annotations that it can get. Some proteins have many annotations, some have far fewer. Ion channels are very important in human disease, so there's going to be a lot of studies, a lot of biological literature about them. And some of these annotations are going to come from text mining. So here's where you see the subcellular localization. And you can see, for instance, there's publications that show. So if you have publications, that's a very important source of information. So text mining comes in there. Pathology, I know some of you are really interested in disease mutations. That's where you're going to get some of that information. More disease mutations and chemistry databases. And down here at the bottom, polymorphism data, that's very interesting. So different allelic variation in a population. Post-translational modifications, or PTMs, is also a very important part of how proteins function. And here's the membrane topology. So SwissPro has labeled, remember earlier I showed you KCNA1 human analysis from TMHMM, which is a transmembrane prediction server using hidden Markov models. So SwissPro has identified these 6 transmembrane segments. There's 1, 2, 3. Here's segment 4. It's the voltage sensor. This is a really hard one because it does have these positively charged residues. And then there's 5 and 6 down here. And you can see on the sequence it's showing you where those positions are, where those membrane helices are. Transmembrane helix prediction, the topology is wrong about half the time. So topology is for a protein. So 1, 2, 3, 4, 5, 6, 7. So for a G protein coupled receptor, they snake in and out of the membrane. They have 7 transmembrane helices. If you missed any one of these, so you missed the 7th one, the topology would be wrong. So it's really easy for the topology, the overall topology, to be wrong. So about half the time topology is right, half the time topology is wrong. And there's different signals for getting topology. So there's a positive inside rule. Which means positively charged amino acids tend to be found on the inside of the membrane. So that's one of the rules that's used. but the topology is wrong about half the time. So the particular location of a transmembrane helix, that might be right, but the topology might be wrong. So in this case, they're telling you where, based on human curation, they determine that the topology is supposed to be. So here's one of my favorite tools that Uniprot gives you, is the ability to cluster proteins, construct an alignment, build a tree, and then view on the alignment where Uniprot has already labeled positions as being membrane, transmembrane, intramembrane, extracellular, and so on. So if you go to the advanced settings, which is what I recommend that you do, you can then choose what database you want to search. So I'm choosing SwissProt, the e-value threshold, the number of sequences you want to see, and here again, that's your query sequence in FASTA format. Even though that looks like it's on 2 lines, it's all on 1 line. Make sure that your header line is all on 1 line. And then you can run BLAST, and you can look at the alignments of sequences, and you can select sequences to include in the multiple sequence limit you're going to get. So I've gone through here and I've selected sequences that appear to align globally, even if there's a partial gap. So this last one here, which is KCNA3 from human, the N-terminus of the hit isn't matching the query, but it's pretty small, so I'm not worried about it having a different domain architecture. I just want to make sure that the proteins effectively have the same multi-domain architecture. And then you can cluster the proteins, and you can ask Uniprot to align them. And you can display on the alignment intramembrane segments and transmembrane segments, and other things as well. I'm particularly interested in the transmembrane helix prediction, because I know how wrong it is much of the time. And here there's like 2 really important points to make. One is that homologs should have the same multi-domain architecture. If they align globally, they should have the same domain structure, which includes the location of the transmembrane helices. It's just like it's exactly protein structure prediction. You know, protein query, the basic idea is you have a query whose in protein structure prediction, you have a protein U whose structure is not known. You want to find a structure using sequence similarity. You align U to S, and if the sequence similarity is high, then you can build a protein structure model using the structure of the hit as a template. So that's called protein structure prediction. And the principle behind that is that evolution conserves structure, mostly conserves structure. Some small changes like lift regions might not be the same, but the most important parts, functionally important parts, will be maintained. Transmembrane helices are a fundamental part of protein's function, so they will be maintained in evolution. All right, so after that little digression, we then look at this alignment. Can you see these positions here? They're not labeled yellow. They agree, the amino acids, with the amino acids shown among their homologs. It's not that they look particularly different to this region. They look very much the same. High sequence similarity in those regions. Ditto for this part, this transmembrane helix. So they should have transmembrane helices in that position, but SwissProt curators didn't annotate those positions as being transmembrane helices. My guess is that the SwissProt curators were just using the results of TMHMM or some other type of prediction tool, where because of the positively charged amino acids that appear, and you can see them here, there's R, 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 and a K. So there's 5 positively charged amino acids that are visible in that region. That makes the hydrophobicity plots problematic. But because sequence similarity is so high, they should have transmembrane helices there. So this is a principle that you can use to improve protein structure prediction and transmembrane helix prediction. And for those of you who are interested in pharmacology and in medical applications of bioinformatics, transmembrane helixes are so important for human and health that being able to improve the accuracy of transmembrane helix prediction is something that is definitely interesting to pharmaceutical companies who are looking for novel drug targets, and just in general. So you can use this concept of using evolution to improve homology relationships, to improve the accuracy, to transfer these annotations along positions. So that was the second concept I really wanted to get to, which I have in my summary slides, which is that you can use not just global similarity to transfer an annotation of function to the protein as a whole, but site-specific annotations, active sites, specificity determinants, post-translational modifications, domain architecture, transmembrane helices. All these annotations can be transferred by homology. So we'll get to that again later, but it's a really important concept. So if it doesn't make sense to you, write it down. We'll get back to it. So annotation transfer. It's a basic concept that we want to get back to, because we're going to use it again and again. So one of the things you can do, if you look at that alignment, go scroll all the way to the bottom, and you can see the guide tree that was constructed by the alignment tool. The last iteration of the multiple sequence alignment is an iterative process. Cluster sequences, build an alignment, build a guide tree to order the sequences alignment in the next iteration. And then that guide tree is re-estimated every time there's a new alignment until it finally converges. And I'm just showing you a subset of the tree. One of you asked about orthologs and paralogs, and I didn't actually put that point on the board. But orthologs and paralogs are all related by evolution from a common ancestor. I think probably the battery on this is a little bit dead. Oh. Possibly. Maybe not. Anyway, these are all KCNA2. So they're rooted here. That's the subtree. That's fish. That's frog. And the rest of these are mammals. So that subtree topology more or less agrees with what we know about the evolution of the species. So that's the KCNA2 subtree. Here's KCNA1, so mouse and rat are together, then human. So that's the KCNA1 subtree. KCNA1 is related to KCNA2 by a duplication event. The duplication event is positioned there on the tree. That means they're paralogs. KCNA1 and KCNA2 are paralogs. They're all related by divergent evolution. Got that? But all the KCNA2 sequences here are each other's orthologs. And that's because when you look at the tree, you can say there's a speciation event that explains the divergence between rat and mouse because it was an ancestral rodent. Back here, that's the ancestor of rat, mouse, and human. And then it goes up. So you're still within mammals here. Now you're getting into frog, and then there's fish. So it's still animals here. So you have distantly related ancestral animals that are on the tree, but they're all speciation events until you get here, in which case you have a duplication event. So when you're predicting orthologs and paralogs, you want to say what's happening at each point in the tree. And if a point in the tree looks like a duplication event, then the groups that diverge from that point are each other's paralogs. So that's basically what's happening is you're doing a reconciliation of the species tree and the gene tree. So then if you're doing this in an automatic way, then you have to have good species trees. And it gets complicated because species trees often have a little bit of mistakes. And the gene trees may not all agree with the species tree. So I don't think I have a good example of that here. Right now, those orders actually agree with species. Yeah?
Could you explain the duplication event versus speciation event again?
Yeah. So if you look at the relationship of rat and mouse, and that node right there that joins these two sequences, that node you'd have to label their ancestor as some ancestral rodent, right? So that's a species relationship. Human, mat and mouse, mouse and rat are joined here by that node, and that's some ancestral mammal. And then you have rabbit being joined in, so it's still mammalian lineage. And then this is, I think that's dog, but maybe it's wolf. That's here. And then Zemla, that's Xenopus levis, so that's a frog. So these are all species relationships. They're all having, you only see the same, you don't see any disagreements with the species tree. That subtree topology agrees with the species relationships as we know them. Until we get up to here, and when you look at the subtree that descends from this group, now we see, that's wrong, we have to go further out here. This side we see mouse, human, and rat. Here we have mouse, human, and rat. So since we have mouse, human, and rat on both sides, there had to be a duplication event. So then at this point in the tree, you have to label that as duplication. So as soon as you have a duplication event on the tree, then the common ancestor between this group and this group is a duplication event, which means they're paralogs. Does that make sense? Good. Any other questions about this? All right, that's good. All right, now string database, and I'm getting close to the end. So the string database, if you type into string the name of a gene, you can use the SwissProt name, as I did for KC-name1 human. You'll get a really nice graphical user interface, and you can click on it and drag it, on any node here. So that's KC-1. You can pull it out and look at it. And it's telling you the edges represent protein-protein associations. There are known interactions, either from curated databases or experimentally determined and colored in different colors. There are predicted interactions from different types of evidence. So I'm going to, since I have some time, I can step you through that. The gene neighborhood, so this is on the genome. So you've heard of operons. Those of you who studied biochemistry, you know that in prokaryotes, that operons on a genome, you might have gene A here, and gene B here, and gene C here. This is on the genome itself. You have 3 different genes, you may have many more, and they're located nearby, which means they're on an operon, and they're transcriptionally co-regulated. So they're all transcribed at the same time because they need to be there for the same biological process or pathway. So if in bacteria, if you have this kind of operon structure, it's the genome neighborhood, gene or genome neighborhood on the genome. And that gives you a clue, a big clue, that they're going to have some protein-protein interaction. Now, protein-protein interaction may either be physical, like they're physically associating, or it might be just more functional temporal. They're not actually physically connecting, but they're participating in the same process. So they're using protein-protein interaction in this more kind of open way. Gene fusion refers to a concept also known as the Rosetta Stone. And David Eisenberg, his lab at UCLA, some years ago, had a series of papers, including the Rosetta Stone method, and protein-protein interaction and things, to assert protein-protein interaction using different types of what they call non-homology approaches. So genome neighborhoods is one. Rosetta Stone basically says if you see in one genome you have the A protein separated from the B protein, which is what's showing up here, and in another genome you have the A gene fused to the B gene, so if they're actually found in one protein, then this form, where they're actually fused into one protein, is a Rosetta Stone that suggests the interaction of A and B separately. So that idea, it's called the Rosetta Stone approach, turns out to be very powerful. And so that's one of the types of tools that they're using here is Rosetta Stone. So it's called gene fusion. Gene co-occurrence is different. So this is like a phylogenetic profile. So we'll talk about that another time. It's not the same thing as a statistical model in SciBlast. It just means if genes are evolving together, then they're likely to be expressed together. That's harder to do well. Text mining, basically you're looking at abstracts. Most commonly, it just says if you see gene A and gene B are their names occur in the same abstract, maybe they interact. So that's an example of text mining. Co-expression. So this is going to be in experimental data. And homology. So if you have, well, that's a little bit different than those orthology relationships. So this gets a little bit more complex. If you're really interested in this, I would say you want to go to the string database, download a paper that they refer to, and read the papers. They list a lot of these proteins that they're predicting to be functional partners. They have scores, and you can see which aspects of the prediction tools were used to come up with that. Text mining is explaining many of them. Homology here, we're really talking about orthology relationships. They're predicted orthologs and other types of data. And that's what you're seeing up here. And so these are my summary comments. I think this is the very last slide, so there'll be time for questions. Or we can go through these summaries slowly. These are the points that I really wanted to make and remind you of the importance. So there's every bioinformatics task has an inherent trade-off between precision and recall. So if you want high precision for homology detection, as an example, you're going to use BLAST. You have very few false positives. If you want high recall, then you're going to have to allow some false positives, but then you're going to have fewer false negatives. So a false negative is something that you should have detected and you didn't. A false positive is something you should have rejected, but instead you accepted it. Everyone understand the difference between false positive and a false negative in homology detection? Are there any questions about that? So BLAST, if you use significant scores, you're going to have high precision and low recall. SciBlast and other types of iterated sequence search methods are designed to improve the recall, but they can have problems with precision. Functions can change even when matches are homologous, and that gets to something I was just explaining earlier about domain architecture not being the same, or having paralogs instead of orthologs. So serotonin receptors are paralogs to dopamine receptors. So you'd want to say it's a G protein coupled receptor, subtype unknown, if you're not sure if they're orthologs and paralogs. How do you predict multi-domain architecture? You can use PFAM, but PFAM will also leave some parts unannotated. So for some proteins, PFAM's just going to say, well, I can explain this part. So the full length of the protein, when you run it to PFAM might say that there is a DUF at this end. And I remember earlier, very few of you had used PFAM, so you won't know what a DUF is. DUF is a domain of unknown function. It's not very helpful in determining function if it's an unknown function. But they cluster proteins based on these domains that they see in many places. They figure out at some point maybe there'll be some experimental data associated with that domain. And then anyone that has that domain, they'll be able to label it part of the protein. But you might have all this that PFAM says nothing about. In which case, what do? You can still use protein structure prediction methods, remote homology detection methods, to improve your ability to classify into domains these regions. In the context of protein structure prediction, one of the first tasks is saying, where are the domain boundaries? So domain boundary prediction is something that X-ray crystallographers often use. So X-ray crystallographers, their job is to solve protein structures and to come up with some ideas about function. But if you have a very long protein, let's say this is 1,500 amino acids long, you can't solve a protein that big. You want to cut it up into pieces of the domain. So then you want to say, we think that the domain boundary is here, and maybe there's another domain boundary here. And the clues for the domain boundary prediction include secondary structure prediction, which has reasonably good accuracy. Per position, you can say it's alpha helix or it's beta strand or it's turn. And that has around 76, 77, 78% accuracy on average. And if you can get the domain boundaries right, then you can try to predict the structure for this piece, or solve the structure for that piece, solve the structure for that piece, solve the structure for that piece separately. And you can actually get those clues by looking at homology relationships. So if when you're clustering homologs using BLAST or PSI-BLAST, your clues for domain boundary could be that you see a lot of local matches here, you see more local matches here, and more local matches here. And that tells you that there's homology relationships that are local, or they're partial, and it helps you see that there might be individual domains in those regions. It's really important to remind you that you need to have the same multi-domain architecture. And as a rough rule of thumb, you want on the order of at least 70% overlap between the query and the hit. What that means is your query protein, if your query protein is 1,000 amino acids long, if you use this approach, which is common, it's not perfect, but if you have a very long protein, then what you're saying is 700 amino acids have to be matched. But that leaves 300 amino acids that aren't matched. And does anyone have an idea of like the typical length of a protein structural domain? Well, let's put it this way. What's the smallest structural domain that you can think of? I'll give you a clue. The defensins that we looked at, the scorpion toxin, those guys are about 50 to, let's say, 60 or so amino acids. You can get a structural domain in 50 or 60 amino acids. So if you have 300 amino acids that are missing, you can have a whole structural domain in there, and that can change the function. So it's not really enough to say 7% overlap, but a lot of the resources that you look at will use it as a rough rule of thumb. If you have a smaller protein, a couple of hundred amino acids, 70% overlap is probably about right. You don't need to go much higher. But just bear in mind that structural domains can occur in smaller regions. Remember, the membrane topology prediction accuracy is low. And then annotation transfer protocols, and I was trying to talk about this earlier. Protein fold prediction, so comparative model construction, also known as homology model construction. Functional sites, you can transfer annotations of active site residues for sure if the amino acids agree. So if you have a protein, the structure, you don't know the structure or the function, but here's your query protein, and here's your hit. If your hit is an enzyme, and it's been experimentally characterized, and they know the active site residues, you can take this residue, which is an active site, and you can transfer the annotation to this residue in the alignment. As long as they have the same residue, if that's a D and that's an R, don't transfer the annotation. But if that's a D, it's an active site aspartic acid, and there's a D in the pairwise alignment, you can transfer the annotation presumed active site by homology. And if you're doing this really well, you would actually include the identifier of the sequence that you use to label it and the E value of the match. So that's how that would work. Transmembrane domains and transmembrane topology can also be transferred, as I was showing you earlier. I think that's the last slide, and it is. All right, so now we have like 6-7 minutes for questions. Thank you. Any questions? Yeah.
You mentioned that trying to solve the structure, like individually of like isomers being
Yeah.
but like the folding of the three-dimensional thing will be completely
Yeah, long protein, and you can only really solve it in pieces. How do you get the conformation of the whole protein together? So you might use electron microscopy to get a rough picture of how the pieces would pack together. That has lower resolution, but in fact it gives you an idea of the packing. But also sometimes you can use comparative modeling, well not comparative modeling in that case, you can use some kind of energy minimization. So you have these pieces, and now you're going to simulate the folding of the pieces against each other and actually use energy functions to say they have better energy when they're in this conformation than in that conformation. So these questions of folding and prediction of folding are things that are dealt with in the CASP competitions where they actually do these global things as well. You had a question back there?
So the string is basically
I can't actually hear you very well. You're saying stream looks at protein-protein interaction?
yeah, so but is it just use evidence from publication? So something.
Stream uses all sorts of evidence. So publications are one source of evidence where they actually have literature that says these two proteins interact. They're also looking at these prediction tools that are like genome neighborhoods. So if there's two genes that are on an operon and they're near each other, then they probably interact. They're looking at the Rosetta Stone method that says in one species they're fused, and so in other species the same genes are likely to interact. So there's a lot of prediction methods going to protein-protein interaction. And Anton, do they have a lecture on protein-protein interaction prediction methods? If you don't, in structural bioinformatics, you'll have a lecture on that. What I can do also is come up with a list of recommended reading for the topics that you're interested in. So for protein-protein interaction, there's a number of really good papers that I can come up with, or also the slides on protein-protein interaction. But it's really part of understanding the processes that a protein participates in, or the pathways. So a lot of that comes from homology relationships. Any other questions?
in terms of
Yeah.
prediction, it's like chaperone folding discriminated between, because it could really mess up, like if you don't know if the protein folds with help of another one.
Yeah, so you're talking about chaperonins and how their role in helping a protein fold properly and avoiding the misfolding. That's what you're talking about?
Yeah.
Well, it's true that all right, so that raises a question of how much you really can do from the primary sequence. So one of the kind of dogmas of computational biology is that all the information that's needed to fold a protein is in the primary sequence. And that's kind of true. But then there's these issues because you do have chaperonins that come in and they help avoid the misfolding. I actually can't say how. I know that prediction methods don't take that into account, and prediction methods are still pretty accurate. So I would guess that what they're doing is that they're helping avoid the misfolding, but in general they don't need that. Everyone good about PsyBlast? You're going to be using BLAST and PsyBlast and PFAM and gene ontology, and you're going to be and SCOP, and you have a homework coming up, right, or some type of a challenge where you have to ask and answer questions of are two proteins related or unrelated, or what is their relationship. So, do you feel like you have what you need to answer those questions? And if you have more questions, you can come down and ask me. Or you can get let out 2 minutes early. All right, you can get let out 2 minutes early.